biological world is comprised of a stunning assortment of organisms. For hundreds of years, biologists have been interested in understanding the patterns that underlie these processes of diversification. So several processes of diversification have been proposed, but today I'm going to focus on ecological speciation and host-parasite interactions. And in particular, we were interested in understanding how do these processes of diversification play out when it's hard to predict where the host is going to be and when they're going to be there. So how do these processes play out in a nomadic system? So simply, ecological speciation, the divergent natural selection arising from differences in the environment. And the environment in this case is comprised of both abiotic factors, such as climate, and biotic factors, such as the interactions within and between species. For example, if we look at Darwin finches, ecological changes, such as a random climatic event, can elicit an evolutionary response in the populations. In three-spined sticklebacks, for instance, limnetic and benthic ecotypes, the fitness of them are dependent on the environment in which they are raised. For our study, we were interested in what happens is there ecological speciation in the red crossbill. So red crossbills are a super cool system for several reasons. The first being they're eruptive and nomadic migrants. Eruptive, and that it's very difficult to tell when the flocks are going to move. Nomadic, and that it's difficult to tell where they're going to move when they do so. So flocks of crossbills may be absent from an area for a decade, invade in hundreds of one year, and never be found in that location again. And crossbills get their name from their distinct crossbill morphology, which is thought to be an adaptation to allow them to access their primary food source of conifer cones. So conifer cones are masting events, and the only way to predict the crossbill movements is based on the masting events of their primary food source. It is important to note that different red crossbill populations have distinct bill morphologies. So here we have 4 different types of red crossbills with distinct bill morphologies. For example, individuals of type 3 have this bill morphology, and individuals of type 5 have this bill morphology. So not only do they look different in a different morphology, but different red crossbill populations eat different food. So type 3 are primarily associated with western hemlock, whereas type 5 individuals are primarily associated with ponderosa pine. And not only that, but different populations make different vocalizations. So for instance, type 3 individuals so they make more of a squeaky tick-tick for their flocking calls. Whereas type 5 individuals have more of a flat and short clip-clip. And these flocking calls are very important in a social context because they allow conspecifics to communicate to each other and say, hey, come over here, we're going to find a new food source. And all these different vocal types can be found in the same geographic location, so in symmetry, and they mate assortatively. So they live in the same geographic location, but they mate assortatively. So in red crossbills then, we see this ecological speciation in that they eat different food, behavioral isolation in that they make different vocalizations, and there's recent evidence for ecological speciation in that different populations are genetically isolated. However, the evidence for this genetic isolation is sparse. Not all vocal types have been investigated, and there have been relatively low sample sizes due to the hard collection methods of finding the species. So my first research question for my master's, I was interested to know, are red crossbill vocal types genetically distinct? And secondly, are red crossbill vocal types, because they have this different ecology and different behavior in their populations, do different red crossbill populations have different parasite communities? To do this, our collaborators at UC Davis sampled 640 crossbills of 4 different vocal types. And just to give us some context here we are, and our sampling locations were in the United States. So if we look more closely, there are 7 different trapping locations in Wyoming, Washington, and Oregon. And so my first question, I wanted to know, if individuals of vocal type 3 caught in Washington, would they be genetically isolated from individuals of vocal type 4 also caught in Washington. And moreover, because of the same ecological factors and behavioral factors, when individuals of vocal type 3 in Washington have similar genetics to those found in Oregon and Wyoming. So to do this, we amplified the red crossbill microsatellite DNA at 10 loci, and this allowed us to determine a coarse red crossbill genetic structuring. And preliminary evidence suggests that some of these vocal types are in fact genetically different. So based on a Markov chain test for population differentiation, we found that individuals in vocal type 3 and vocal type 4 were significantly differentiated. And this is really interesting because vocal type 3 and 5 individuals are the most morphologically distinct. And keep in mind that these vocal type 3 and 5 individuals were caught in the same geographic location. So now, turning to other processes of diversification. So if we have this ecological speciation in the system, well, how does this play out with the host and parasite interactions? How do red crossbills interplay with their blood-borne parasites? And so it has been proposed that infectious disease is an important driver behind patterns of biodiversity we see across latitudes. So in general, as we go to lower latitudes, we see more diverse species communities. And it is thought that this may be because also at these lower latitudes, there are more diverse parasite communities, which then allow local adaptation in their parasites and therefore diversification. We have to keep in mind, however, that parasites are not limited to one geographic range. Parasites are incredibly widespread, and they account for approximately 40% of all described species. They're taxonomically diverse as well. There are parasitic bacteria, parasitic fungi, parasitic plants, and even some parasitic vertebrates. And just in this session, we've seen that the effects that parasites have on their individual hosts have been well characterized. We even know that the effects that parasites may have at the population level are well characterized as well. But hosts may also influence parasite evolution. So parasites may speciate in response to host speciation, and that host speciation may actually induce parasite speciation in a process known as co-speciation. So a simplistic representation of that is the matching phylogenies of the parasite and the host in this process of co-speciation. The outcome, so the way that these host-parasite interactions shape the evolutionary trajectories of both populations depends on many factors. The ecologies of the host, for example, so in this system, the red crossbills, the ecologies of the parasites themselves, and in my system, the ecologies of the vectors. So we were interested in avian humanozoan parasites, which infect approximately 75% of all described bird species. The first two genera were sister taxa, Plasmodium and Hemiproteus. The third and final genus we were interested in was Leucocytozoan. It is important to note that Plasmodium and Hemiproteus are indistinguishable by morphology alone. And I'm going to focus on Hemiproteus and Leucocytozoan because we found no Plasmodium infections in our red crossbill samples. All three of these genera are intracellular protozoa and belong to the avian complexa. So just as the parasites themselves are diverse, so too are the vectors and the way in which they're transmitted. Hemiproteus are transmitted by louse flies and biting midges, whereas Leucocytozoan is transmitted by black flies. So as you can imagine, if they're thought to infect 75% of all bird species, these parasites have been extensively studied in their avian hosts. So we see examples in the literature of birds that live on islands, birds that have very defined geographic ranges, and different populations of birds over continental ranges. But many of the studies exploring the host-parasite coevolution in avian humanozoan parasites have looked at systems with predictable geographic ranges. So the bird hosts, we can predict where they're going to be and when they're going to be there. So this begs the question, what happens in systems like the red crossbill? Bringing me to my second and final question, are both red crossbill vocal types subject to different parasite communities? So to do this, we amplified a portion of the parasite mitochondrial cytochrome B. We sequenced the resultant amplicons and constructed a parasite phylogeny. So we found actually a low overall amount of infection was detected. So there are only 48 birds infected out of 640 with this molecular method. There are 20 co-infections. So I'm currently working on cloning to tease apart these co-infections. So today I'm just going to talk about the samples that were not co-infected, so 24 proteus infections and 4 leukocytosomal infections. So based on the maximum likelihood, here we have 2 distinct lineages of proteus and 2 distinct lineages of leukocytosomal. And so the main takeaway from this slide here is I want you to focus on the first proteus lineage. We only detected this in individuals of type 3, which is very interesting. And these type 3 individuals came from all 7 of our sampling locations. If we then look at the second lineage of proteus, we see that we isolated this lineage in type 5 individuals, type 4 individuals, and type 3 individuals. So even though we have low sample size and we're trying to increase that by cloning, there might be something interesting going on. So just another representation of this data here, we see type 3 individuals. Over half of the infections in type 3 were the specific lineage of proteus. And in type 5, we don't see any indication of this lineage in type 5. So even though our sample size is low right now, there's still indication that there might be specificity going on in the system, at least with the vocal type 3 individuals. So just again to represent this on the map, individuals of vocal type 3 caught at all these locations shared the same proteus lineage, but then other vocal types caught at these locations, we didn't find it there. So to conclude and to summarize, in this nomadic system, there are different processes of diversification. We have evidence of ecological speciation, and we have evidence that the host-parasite interactions may also be causing diversification in the nomadic system of the red crossbill complex. So with that, I would like to acknowledge my fleet of bird nerds at the University of Western Ontario, my collaborators at UC Davis, the funding bodies that got me here, and you guys for sticking it out in the last session. Thank you very much. The vector. So we don't know which species of each vector carries the certain lineages, because we weren't allowed to get to the morphospecies of the lineage. But they're pretty widespread. So I know our lab also studies song sparrows in northern Ontario, and they have the same vectors there as well. So we kind of think it's driven by the oviposition site of the vectors.
So it could be that just type 3 was exposed to the vector more than type 5?
Exactly. So the microecologies, they're primarily associated with one conifer host, but potentially that species is maybe near more still water, and then the oviposition of the vectors might be near still water as well, and then they're near the emergence of the vectors, and then they might be infected that way.